Proteins do an astonishing variety of things. The muscles that enable you to move are protein, but so is that hair on your head. The carrier of oxygen to your tissues, protein. The messengers that keep blood glucose levels in check, protein. The enzymes that build and break down molecules in cells, protein. And that's just a tiny fraction of the things that proteins do. In the last lecture, we learned about the 20 amino acid building blocks that cells use to make proteins. Each amino acid different from the others because of a unique R group. We also saw that amino acids are linked together by peptide bonds. When molecules have many peptide bonds, they're called polypeptides. How can polypeptide strings of amino acids do all of these things? In this lecture, we'll take a look at the unique structures that chains of amino acids take on, allowing them to carry out such a wide variety of specific functions. The English language has 26 letters in its alphabet, and we can construct every word in the language using different combinations of the letters. So it's easy to imagine that 20 amino acids can, likewise, be used to create many different protein words. When we write words, we know which letters we use, and the order of the letters matters. My name is Kevin. I have a friend named Keith. Though both of our names have 5 letters and start with K-E, Kevin is not Keith and never will be. Likewise, which amino acids are used to build a protein and what order they are in distinguishes one polypeptide from another. A polypeptide will have at least a sentence worth of letters. And even a chain of 100 amino acids is, by cellular standards, quite small. That's the protein equivalent of a tweet worth of letters. A medium-sized protein like collagen, which is the most abundant protein in our body, has the number of letters in a short speech like the Gettysburg Address by Abraham Lincoln. The largest known protein is a muscle protein called titin, and it has over 30,000 letters. That's roughly the number of letters it took to write this 30-minute talk. Protein letters differ from letters in a sentence, though. Letters and proteins are physically linked to each other by the formation of peptide bonds between the alpha carboxyl group of one amino acid and the alpha amino group of the next. These very strong covalent bonds leave a free amino group on the first amino acid and a free carboxyl group on the second one. The carboxyl group is free to make another peptide bond. Polypeptides grow in this way by addition of new amino acids onto the free carboxyl ends. This scheme results in a chain where the amino acid on one end has a free amino group, while the amino acid at the other has a free carboxyl group. The end with the free amino group is called the amino or N terminus. The other end is called the carboxyl or C terminus. To make it easier to keep track of amino acids in a protein chain, we assign single letters to correspond to each amino acid. So it's literally a sort of amino acid alphabet, as I said. About half of the letters are obvious because they're the first letter of the amino acids' names. But there are 4 amino acids that start with A, 3 start with G, 2 start with L, 2 start with P, and 3 start with T, so we do have to make some adjustments. You do not need to learn these abbreviations, but feel free to pause and study them, either here or using the supplementary materials, if you're curious. So if the word Kevin represented a polypeptide chain, what would I be? Well, the first 2 are tricky. K stands for lysine, go figure. E stands for glutamic acid. And then VI is valine isoleucine. And lastly, N is asparagine. You can probably see where this is headed. Since each amino acid has slightly different chemistry than all the others, changing the order and composition of the amino acids also changes not only the words that they make, but also what the polypeptide chain does and how it works. Nivek, my name spelled backwards, is certainly not the same as Kevin. That is your first lesson in protein structure. Proteins are made up of chains of amino acids that vary in length, the kinds, and the order of their constituent amino acids. This sequence of amino acids is referred to as the primary structure of a protein, and that determines its properties. Two different proteins will never have the same sequence of amino acids, but every molecule of a given protein will be identical. For example, every molecule of the digestive enzyme trypsin has the same order of amino acids as every other trypsin enzyme. But just like the sequence of words alone does not make a novel, a sequence of protein letters does not make a functional protein. Like the elements of a plot must work together properly for a novel to be enjoyable, interactions between amino acids in a protein must occur for a protein to function. These interactions give rise to a protein's characteristic structure. This structure is so important to function that when a protein loses structure, its function is lost too. We've already seen some clues about how this works. As you know, amino acids have different side chains or R groups. The chemistry of the R groups affects the way any two amino acids interact with each other. The positively charged R group is attracted to a negatively charged one, creating a force holding these two groups together. For example, at physiological pH, lysine's R group is positively charged and is attracted to the negatively charged R group of aspartate. Conversely, any two groups with the same charge are pushed apart by repulsive effects. Charges, though, are not the only kinds of interactions affecting structure of proteins. The hydrophobic R groups all try to hide away from water. Let's remember the analogy of a middle school clique where the kids excluded outsiders. The same thing happens in a protein with hydrophobic side chains, which associate with each other and exclude water. In a protein, this tends to occur inside of a protein, as we shall see, and it's a driving force for the protein to assume its functional shape. The other amino acids are hydrophilic and are more likely to seek and interact with water because their R groups can form hydrogen bonds. R groups aren't the only parts of a protein where interactions occur. Even the double-bonded oxygens and amine groups on the peptide backbone get into the act. This is why the order and location of the amino acids in a protein are so important. They determine what interactions can occur and ultimately the shape of the protein. So amino acids all by themselves establish the structure of a protein. We give the process of establishing protein structure a name. It's called protein folding. Protein folding is complicated and occurs at multiple levels, depending on how far apart in the primary sequence amino acids are that are interacting. Let's start our discussion of protein folding with interactions between amino acids close in primary sequence. These give rise to what we call secondary structure and occur mostly as hydrogen bonds between atoms in the peptide backbone. Specifically, these involve the C double bond oxygen of one amino acid and the nearby NH group of another. Such hydrogen bonds can, depending upon the sequence of the polypeptide in the region, give rise to two main types of characteristic helical structures, plus two lesser variations. The alpha helix structure is one such secondary structure. It was originally predicted by Linus Pauling and Robert Corey, for which Pauling was awarded the Nobel Prize in Chemistry in 1954. Let's take a look. There are two image formats we commonly use to depict proteins in biochemistry. A ribbon diagram is a simple cartoon depiction of the structure of a protein. It guides our eyes to depict the secondary structures within it. In a large structure like a complete protein, ribbons for guiding our eyes are helpful because the enormous collection of atoms looks like a giant tangle, and it is hard to discern the underlying patterns. To help you to see the forces that help stabilize an alpha helix, I've superimposed the individual atoms of the polypeptide's backbone onto a ribbon. The stabilizing forces are shown as dotted lines. Each of these dotted lines is between a hydrogen and oxygen, similar to what we saw between molecules of water. Yes, those are hydrogen bonds. The hydrogens are located on NH groups in the polypeptide backbone, and the oxygens are on the carbons of a C double bonded oxygen group on the backbone for a different amino acid. Pauling's alpha helix is a three-dimensional coil. If we look at it from the top, instead of looking at it from the side, we can see that the R groups are all oriented so as to point away from the central core of the helix. R groups can have a large influence on the stability of a secondary structure. Their size and flexibility influence the type of secondary structure a given sequence assumes. Amino acids with large R groups, such as tryptophan, histidine, or phenylalanine, have spatial limitations. They won't fit into the small spaces like those of glycine or alanine. Proline is the most inflexible amino acid, and its rigidity is also an important consideration. A second structure Pauling predicted is what has been sometimes described as a flattened helix, a 2D pleated structure called a beta strand. It turns out a beta strand readily form hydrogen bonds with other beta strands to give rise to higher order structures called beta sheets. Alpha helices and beta sheets are the two most common secondary structures, but there are also variants. The alpha helix has 18 amino acids per 5 turns of the helix, which works out to 3.4 amino acids per turn. But there are also variants called the 310 helix and the pi helix with slightly different numbers of amino acids per turn. Both 310 and pi helices only occur over very short stretches of amino acids. Another consideration is the way a helix coils. There are two possibilities, referred to as right and left-handed helices. The easiest way to determine the handedness of a helix is by looking at the direction of the coil moving away from the viewer. Looking end-on, a right-handed helix will have coils extending away from the viewer in a clockwise direction. A left-handed helix will coil counterclockwise. This phone cord is left-handed. This staircase is right-handed. In proteins, right-handed helices, that is, rotating clockwise away from you, are far more common. As I noted earlier, the different R groups of the amino acids in a stretch of polypeptide play important roles in determining secondary structures. By examining the known structures of thousands of proteins and tallying how often each amino acid is found in the different forms of secondary structure, one can determine a likelihood of each amino acid being in any of the structures. This data shows only tendencies, as there are no absolutes for finding any amino acid in any particular secondary structure. Indeed, as we shall see for the fibrous proteins, some amino acids in their secondary structures go counter to the values shown in this graph. So let's talk about fibrous proteins. These are proteins that are comprised mostly of primary and secondary structures, sometimes combining into bundles making long, strong fibers. Some fibrous proteins contain only a subset of the 20 amino acids organized in a regular repeating pattern. For example, silk is a fibrous protein called fibrin that uses the 3 smallest amino acids to make beta sheets over and over and over. When you compare fibrin's repeating sequence of amino acids to the data we saw about which are more likely in a beta sheet, you will notice that its amino acid composition of glycine, alanine, and serine isn't what you might expect, as I noted earlier. Nevertheless, fibrin is rich in beta sheets built with repeated sequences of these small amino acids. The structure of silk fibers gives them very high tensile strength, and woven silk is one of the most durable of fabrics. Ancient silk garments have been found at archaeological sites. The strength, softness, and sheen of silk make it a much sought-after protein. This extraordinary garment was made using the beautiful silk of the golden spider, which was on display at the Victoria and Albert Museum in London in 2012. A garment made entirely of spider silk. You may recall from the last lecture that cells use L-amino acids almost exclusively to make proteins, and silk protein is no exception. It turns out that with age, some L-amino acids in proteins can gradually flip into the D configuration, until it reaches the 50-50 mix found in non-biological amino acids. This non-biological process occurs so slowly, with a half-life of 2,500 years for L-aspartic acid to form D-aspartic acid, that it allows scientists to determine the age of silk found in ancient sites. Fibrous proteins are grouped into several large superfamilies, some of which contain numerous examples. The keratin family, for example, is broadly subdivided into alpha-keratins, which contain abundant alpha helices, and beta-keratins, which are rich in beta sheets. Alpha-keratins make up hair and the outermost layers of our skin, as well as the horns, nails, hooves, and claws of other mammals. Beta-keratins are commonly found in reptiles and birds and are the hardest keratins, making up shells, beaks, scales, nails, and claws. The keratin in hair has mostly alpha helices and is rich in the amino acid cysteine. Disulfide bonds between individual alpha-keratin fibers make possible fiber bundles that give hair its strength and also its curl. Perhaps the most important fibrous protein for multicellular organisms is the superfamily known as collagen. Collagen is in the extracellular space of connective tissues and makes up 25 to 30% of all the protein in our bodies. Because it functions to hold cells together, collagen has been described as the body's glue. Now, if you've ever eaten gummy bear candies or Jell-O, you've encountered collagen whose triple helix has been heated and denatured to form gelatin. Several aspects of collagen structure are unusual. First, it contains a triple helix of 3 left-handed polypeptide chains called procollagens, formed in a unique arrangement. The weird part is, next, these left-handed chains are braided together in a right-handed fashion to make a tight bundle called tropocollagen. The tight packing of the chains leaves little space in the central core of the fiber. Such a structure is possible due to collagen's abundance of glycine, which has the smallest side chain of all the amino acids and facilitates tight packing with its hydrogen R group pointing to the center of the helix. This is why glycine is in every third position of collagen's amino acid sequence. You can also see a great abundance of the amino acid proline and in a modified form of it called hydroxyproline, where it's labeled here as HPR. Recall that proline's ring side chain limits flexibility. Hydroxyproline is formed when hydroxyl groups are added onto proline rings after collagen is made, and it has the same rigidness as proline. A similar hydroxylation is made to the amino acid lysine in collagen to create hydroxylysine. The hydroxyl adding reactions to lysine and proline both require vitamin C, which is one reason we need vitamin C in our diet. Hydroxylation of proline is important for giving collagen thermal stability at the temperature of the body. As body temperature increases, so too must the amount of hydroxylation. For example, fish are cold-blooded, so they have fewer hydroxyprolines in collagen than we do. The hydroxylysines differ from ordinary lysines in that they react with each other to form covalent bonds, joining different triple helical tropocollagen bundles firmly together. Such bonding has the effect of improving collagen strength considerably. Decreased hydroxylation of lysine and proline, which can happen when there's not enough vitamin C, results in unstable, weakened collagen. This leads to a condition known as scurvy, which was the cause of death for as many as 2 million sailors up until the 18th century, when James Lind, a physician in the Royal Navy, showed that sailors who ate citrus fruit rich in vitamin C did not develop scurvy. It's the reason sailors got the nickname limeys. So the regular repeating secondary structures of collagen and other fibrous proteins are very important. Most proteins are not fibrous, and in these proteins, secondary structures do not extend for long distances as they do in fibrous proteins. Instead, for non-fibrous proteins, secondary structures are local patterns connected by bends and folds between the local secondary structures to create what is called tertiary structure. The most distinguishing structural features of non-fibrous proteins are called turns, of which the most common is known as the beta turn. A beta turn essentially reverses the direction of a polypeptide chain when drawn in 2 dimensions. Common amino acids found in beta turns are proline and glycine. Such a turn is stabilized by, you probably guessed it, a hydrogen bond. Interruptions like turns lead to a more complex 3-dimensional structure than a simple alpha helix or beta sheet. Another non-repeating feature of polypeptide chains is the random coil. Random coils are short, random-looking stretches of a polypeptide. Like turns, random coils interrupt repeating secondary structures and allow the polypeptide chain to, again, fold on itself. Bends and random coils provide enormous diversity for the shapes and structures of folded proteins, all of which started out as an amino acid chain. It is this diversity of form that allows proteins to perform their very specific functions. You can see thousands of known protein structures in the online Protein Data Bank, also called PDB. Proteins that fold into tertiary structures are called globular proteins, and they are the most abundant proteins in cells. For a tertiary structure to exist, there must be forces to stabilize it. In contrast to what we saw with secondary structure, where the stabilizing forces were hydrogen bonds, tertiary structures are stabilized by several different forces. Most of these are not covalent bonds, but rather weaker interactions between amino acids in a protein sequence. Let's take a look. Like secondary structure, hydrogen bonds form and help hold the structure together. Almost as strong as covalent bonds, ionic bonds between positively charged R group of one amino acid and the negatively charged R group of another are stabilizing forces. Most distinctive, hydrophobic interactions are major considerations in tertiary structure of most globular proteins. Hydrophobic side chains of molecules tend to associate with each other and to exclude water, as we have seen. As a result, in an aqueous cellular environment, polypeptides tend to fold so that the hydrophobic amino acids are buried in the interior of a globular protein. This allows their water-fearing R groups to cluster together and avoid water. The tendency of hydrophobic amino acids to behave this way is called the hydrophobic effect, and it plays a very important role in protein structure. Conversely, the amino acids with hydrophilic R groups tend to orient on the outer surface of a protein where they can interact with water. Many proteins also contain metal ions. A single metal ion can interact with the R groups of multiple amino acids by what is called coordination. Metal coordination most commonly involves the R group nitrogens of histidines, the sulfurs of cysteines, or the carboxyl groups of aspartates. The most common metals found in proteins are iron, zinc, and copper. Iron exists in numerous forms in cells. In its role as an electron carrier, it is commonly complexed with sulfur atoms and sulfur in the side chains of cysteine. Finally, a significant stabilizer of tertiary structure is the covalent disulfide bond. As you learned in the lecture on amino acids, the side chains of cysteines contain SH groups, which can give up their hydrogen and form a disulfide bond with a partner S from another cysteine that has lost its hydrogen. When cysteines in different parts of a polypeptide are brought together, as can happen during the folding process, disulfide bonds can form readily. Cysteine has the only side chain commonly forming these strong covalent bonds. So why do we care about forces stabilizing tertiary structure? The answer is that a protein's function depends on its structure. If a protein loses its 3D structure and undergoes what in biochemical terms is called denaturation, its structure is changed and its function is lost. That's what happens when we cook things. Proteins get denatured because the heat overcomes the forces stabilizing 3D structure. Forces that help a protein to maintain its structure are therefore pretty important. Let us zoom in on tertiary structure a bit to understand related structures, sometimes called supersecondary structures. Human cells can make about 20,000 distinct proteins, but there are many variations on that core group, allowing us to make well over 100,000 different forms. The vast majority of cellular proteins are globular, folded proteins. Each protein has a unique amino acid sequence and therefore a unique 3D structure. But that is not to suggest there are not commonalities among proteins. The hundreds or thousands of different models of cars sold around the world share common features like tires, engines, and doors. Similarly, proteins often have structural subassemblies or domains that carry out functions common to different proteins. For example, the protein in our blood called albumin has 3 main domains. A protein domain is a region of typically 50 to 250 amino acids, usually contiguous, that can fold independently into a functional unit within a larger protein. Let's consider a protein domain known as EF hands. The function of an EF hand domain is to bind calcium, and it appears in many proteins binding calcium. Binding calcium requires a precisely organized set of amino acid side chains whose arrangement in 3D space optimizes holding on to the calcium ion. This is a calcium binding motif. A motif is a substructure of a domain that performs the function of the domain. For the EF hands domain, the calcium binding motif performs the domain's function of binding calcium. In general, a motif is a substructure within a domain, while a domain is the larger surrounding protein structure supporting the motif. Finally, there's one more level of protein structure for us to consider. The structures are more complex, but the idea is pretty simple. Proteins that are made up of more than one polypeptide are said to have quaternary structure. Hemoglobin, for example, has 4 folded polypeptides that associate with each other to form the functional protein. Note that these are not domains within a single chain. These are 4 distinct polypeptide chains interacting with each other, but otherwise they're not physically connected. Okay, let's wrap this up. So 20 amino acids can form an almost unlimited number of protein words, similar to our language. The spelling of each protein word matters, but even more important for protein function are the structures they form through a process called folding. Secondary structures like helices and beta sheets arise through interactions between atoms in the polypeptide backbone. Tertiary structure comes about by the folding of polypeptide chains into a more complex and compact globular form. The final level of protein structure is quaternary structure. Only proteins composed of multiple subunits have quaternary structure, which is defined by the interactions among the individual folded polypeptides. But quaternary structure can help regulate the activity of proteins, as we will see in numerous examples through the rest of this series. The 20 amino A's define a protein many ways. Their order in a peptide chain determines forms that proteins gain. And when they coil, it leaves me merry, because that makes structures secondary. It's tertiary, I am told, that happens when a protein folds. And if the chain's not solitary, they have a structure, quaternary. They're nature's wonders, that's for sure, creating problems as well as cures.